The adult form of any organism is maintained by a flow of materials, so that the substance of which the organism is made is continually changing, but the form remains constant. We're going to consider the mechanisms that allow cells to differentiate and interact to produce the familiar and complex shapes that we recognize as an adult organism. This includes the process of morphogenesis. Morphogenesis is the generation of shape or form. These microscopic slime molds and the hydra are simple organisms, which we'll look at in detail later. But we'll start by looking at morphogenesis in the toad, a higher organism not dissimilar in its early stages from ourselves. Speeded up 500 times, here are the early cleavages of an amphibian embryo. After 10 hours, there are several thousand cells, all apparently identical. There's no overall growth in size of the embryo. During gastrulation, cells stream inwards. A dark line appears, the outline of the blastopore, through which the cells move. Inside, they will form the wall of the gut and the mesoderm. At the same time, the embryo starts to elongate as cells create a central axis. A trough forms, and the edges of the trough close to form a tube. This is the neural tube, from which the entire nervous system will develop. The anterior end will be the brain, and all the rest forms the spinal cord. A recognizable tadpole starts to emerge. From this stage on, the animal feeds and grows. During metamorphosis, these tadpoles will absorb their tails, develop limbs, and become adult toads. The toad has a much more complex shape than our next example, the hydra, but the same principles that govern the generation and maintenance of its form apply to any organisms. So we can use the simple microscopic creature hydra to illustrate these in a more simple way.
Most animals, when they develop during their embryogenesis, are growing, and they grow through their early stages of adulthood and then possibly stop growing. Some of their tissues may continue to grow, but the form is essentially static. Hydra is very different in this respect, in that it's much more dynamic. The tissues, all of the tissues, grow throughout the lifetime of the animal. Now, you might think that this should lead to the animal growing larger and larger throughout its life, but it doesn't. It's constant in size for a simple reason. It's continually losing tissue. At the same time, it's producing it. It's in a steady state. A consequence of this is that all the cells are continually changing their location.
Some are displaced towards the head and up the tentacles from the body column. Some are displaced towards the foot. In fact, it's much like looking at a fountain in which you have a constant form, but it's very dynamic. Depending on their position, some of the cells which move downwards in the body column can grow to form a bud, which will become a new hydra.
In hydra, there's about 15 cell types, and many of them have specific regional distributions, such as mucus cells being located in the head. And you could imagine with this continual flow of tissue that they would just be lost. That's not the way it works. Their cells are continually replenished. And to give you an example how that works, we can consider the epithelial cells of the ectoderm.
The hydra in longitudinal section can be seen to be composed of two layers of cells, the epidermis or ectoderm and the gastrodermis or endoderm. The body is a simple sac with a mouth at one end surrounded by tentacles and a basal disc or foot at the other. Some of the cell types are shown here in this enlarged section through the body wall. Their position around the body of the hydra can be seen more easily in this cross-section. The epithelial or interstitial cells are the simple undifferentiated cells. But what happens to these cells?
On the body column, these epithelial cells are displaced either down into the foot, where they become gland cells, or up into the tentacles, where they become battery cells. It's a motion where they move up and onto a tentacle. A battery cell is an epithelial cell which has a number of cells embedded within it, each with a small hair, and hence it looks like a battery of guns, and that's how it got its name. And in this process, these cells, the battery cells, will be lost off the end of the tentacle by tissue movements, and they're replenished because they're converted from epithelial cells into battery cells. This maintains this population, and this is true of several of the populations. So this is how the pattern can be maintained. Since these cells are continually changing their location, they must somehow have information about their location, or the cells, as said in as sort of simple terms, as they read their position. And if they're reading their position, they must be getting it from outside. This normally means interactions with other cells, and logically one can think of two extremes. You talk to your cells who are your immediate neighbors, or you might interact with cells that are far away through some sort of global process. Now in Hydra, we have a fair amount of information suggesting that most of these interactions are global.
It's important to understand these global properties. How are they investigated in Hydra?
Well, one thing that's really nice about Hydra is it is amenable to several kinds of experimentation that allow you to move cells around, change their location and change their neighbors, and ask what happens to them. One of them is built into its well-known capacity for regeneration. If you cut the head off of a hydra, it regenerates it. If you cut the foot off of a hydra, it regenerates a foot. If you cut out a piece of a body column, it regenerates a head and a foot. And you have a normal animal, just smaller.
And to summarize, if you cut out a section from the body column, all three sections will grow into complete animals, their size depending on the size of the section.
The kind of regeneration that Hydra undergoes, and this is important, is a form called morphollaxis. In this form of regeneration, the animal or the cells, for instance in head regeneration, the cells at the apical end simply reorganize to form a head. There's no cell division involved, there's no cell growth. Counter to this is, for example, in amphibian limbs, if you cut off a limb, it regenerates the limb, but the tissue must divide and the whole limb must grow for the regeneration to take place, and you get a normal-sized limb. The value in this method is the following. If you cut a hydra about in the middle of the body column, the remaining lower end will regenerate a head. And what's happened is that some of the epithelial cells here that were in the body column are suddenly moved into the head and suddenly become battery cells of the tentacle. This gives you an explicit experiment showing you that these cells are reading their position. So any part of the body column can form either a head or a foot. Now, there's another interesting aspect to this one has to keep in mind. If you take a piece of the body column out, the apical end will always regenerate a head, and the basal end will always regenerate a foot. The tissue knows which way's up, or we refer to this as the polarity of regeneration. And so the question then is, what's the basis of this polarity? Now, there's an additional question one can ask, which probably has occurred to you in the meantime, and that is, that if any part of the head, any part of the body column can make a head or foot, why doesn't it do it? Why doesn't it just go ahead and do it? The answer to both of these questions comes from a kind of experiment that was first carried out near the turn of the century, and it's a transplantation experiment. If you take a piece of tissue from the head region and implant it into the body column, it will always induce a secondary axis. That is, the tissue will grow out and you'll find a head at the end of it. Now, you can also do this experiment by taking a piece of tissue from the body column and implanting it elsewhere in the body, and you'll get the same result, but it'll be a much less frequent result. It may happen in about 20% of the cases. These kinds of experiments were formed extensively in the 60s and even more so in the 70s, and they established the kinds of processes that were involved in this polarity of regeneration. Turns out it was a pair of developmental gradients that is responsible, or generally responsible, for the head forming at the apical end. The two kinds of gradients, one is head inhibition, the other is head activation, and they're easy to describe.
The graph shows the difference in the activity of the head activator substance, known as morphogen, in the three main regions of the body. Activity is measured in biological units, the maximum being in the head and minimum in the foot. The head inhibitor has a similar pattern of distribution.
If you take a piece of tissue and implant it in the side of a normal animal, it will make a head in about 10% of the cases. If you implant it into a host in which you've removed the head, the implant will make a secondary axis in about 75% of the cases. The conclusion is that the head was inhibiting the implant from making a secondary axis. If you take a piece of tissue of the body column near the head and transplant it near the foot, it will make a head in 50% of the cases. If you take a piece of tissue from far down the column and transplant it near the foot, only about 15% will make heads. That is, the ability to make a head is also graded down the column. Now, this is, unlike head inhibition, is a property of the tissue, and it's stable. Many more experiments have established the dynamics of these two gradients and how they interact during regeneration. And a very simple or simplified version of the overall picture that we have of that now is that if you decapitate a head, decapitate an animal, there is no more inhibition coming from the head. Inhibition in the body column vanishes, and then head activation everywhere begins to rise. Because the head activation level, as measured by induction, is highest at the apical end, it rises to a head level very quickly there, establishes a head, produces inhibition, prevents the rest of the body column from trying to make a head. And in that way, you've reestablished a head there, and no other part of the organism will make a head. Of course, in addition to the two gradients that control head formation, there's a similar pair that controls foot formation. These have similar properties. It's just that their maxima, instead of being in the head, are in the foot. Now, a critical question, knowing that these gradients exist, is we want to understand both the cellular and molecular basis of these gradients. It's sort of the next level of understanding of the process. We have a fair understanding of the cellular basis, and we're getting inklings of the molecular basis. A number of experiments involving mutants and abilities to make chimeras between mutants and normal animals have established that the cell types responsible for generating, maintaining, transmitting the gradients are the epithelial cells. Neurons may play a role in a modulatory way but not in a main way. And those are the only two cell types that are probably involved. The molecular basis of these gradients is less well understood, though we have some inklings as to what might be going on. One of the problems is, how do you investigate this? What do you look for? This has been a classic problem in all patterning work because people have had an idea as to what they're looking for. They think there are molecules called morphogens, which are responsible for initiating a process that will lead to a spatial pattern of differentiated cells.
Knowing about cell interactions and the role of activators, inhibitors and polarity, what would you predict would happen to Hydra if we cut out a section of body column, mash it up so that the cells disaggregate, and then these cells are centrifuged to form a pellet? Watch what happens. You can see that polarized structures emerge and develop into new animals. So what do we now know? We know that cell interactions are vital in morphogenesis, and we know that polarity arises spontaneously from these interactions in Hydra. Similar processes occur in other organisms as essential parts of their life cycles, and we'll use the slime molds to illustrate this. The slime molds, in the vegetative phase of their life cycle, exist as individual amoebae, which are non-polarized cells that move about and engulf bacteria, growing and dividing as autonomous individuals. They pay no attention to one another. When food supplies run out, their behavior changes dramatically, and they enter into a cooperative form of interaction. They start chemically signaling to one another, moving towards the signals so that they aggregate. The chemical responsible for this is cyclic AMP. The cells give off a burst of cyclic AMP, which then diffuses, and neighboring cells are stimulated to produce a pulse of chemical. So the signal propagates from cell to cell. In response to the signal, the amoebae send out a pseudopod in the direction from which the signal comes, and so move towards the source. The waves of response and movement can be either concentric circles propagating from the center, where the initiator cell is located, or the signal can also propagate with a spiral motion. At the center of such a spiral is a circle of cells that are stimulating one another, and the signal propagates out from this circular source as in a Catherine wheel. Since this is time-lapse, we're seeing everything speeded up. The actual frequency of the signaling is about 1 pulse of cyclic AMP every 7 or 8 minutes. The rate of propagation of the signal is about 5 microns per second. Once the cells have become sticky and joined together, the coherent whole subsequently transforms into an organism of characteristic form consisting of different cell types. This strange-looking creature is known as a slug. Although microscopic, it contains several thousand cells. In this stage, the organism is not feeding, it only feeds in the amoeba stage. As the slug moves, it leaves a slime sheath behind it, hence the name slime mold. The slug migrates continuously over the surface of rotting vegetation, which is its natural habitat. During this period of migration, 2 types of cells differentiate. They are known as the pre-stalk and the pre-spore cells. The pre-stalk cells are at the front of the organism and constitute about 1/3 of the slug. The other 2/3 are the pre-spore cells. Under certain circumstances, slugs separate into parts, but even so they will still reform into polarized structures, taking on the same pattern of pre-stalk and pre-spore cells as the original slug. After a period of migration, the slugs stop moving and settle down into a form that's called the Mexican hat form, due to its strange shape. The stalk cells then form a column up which the spore cells move. At this stage, the two cell types become obvious, and they separate from one another to form the structure known as the fruiting body. The little hiccup that occurs when the mass of spores changes its shape from an ellipsoid to a sphere is due to a transition from a state in which they adhere to the stalk to one in which they cohere with one another. The spores are resistant to desiccation, and when moist conditions occur, the spores are released and the amoebae then re-emerge and start the life cycle over again. The spores on the fruiting body facilitate the distribution and spread of the organisms in their habitat. The stalk and spore cells differ in gene expression, proteins, and metabolic state. The stalk cells lose their nuclei during final differentiation in the stalk, developing a hard cell wall. So only the spore cells survive to produce the next generation. This is a good example of cooperative activity in which only some of the initial amoebae survive to propagate the species. Different species of slime mold have different forms, and some of these have multiple fruiting bodies. These are similar to multiple-headed hydra that arise from pelleted aggregates of disassociated and randomly mixed hydra cells. A very similar experiment can be done with a slime mold slug by physically mixing the cells together and then letting them reorganize. The result is the formation of several slugs that move off in varying directions. In the hydra case, the initial polarity of the organism was destroyed by the random mixing of the cells. But in the aggregate, groups of cells spontaneously produced polarized structures that developed into heads and bodies. The slime mold starts off as an aggregate of cells which spontaneously generate polarized structures with either a single or many axes, depending on the species. So presumably, similar types of processes are occurring in both hydra and the slime mold, despite their great taxonomic differences. Morphogenesis, pattern formation, and maintenance of form is a dynamic process that depends upon the interaction of cells and the formation of polarized structures. We have illustrated this with 3 organisms, but it equally applies throughout all organisms in the living world.